It's important to note that, the kind of experiments we've done are relatively straightforward. What I mean by that is that it's easy to understand, when you explain to somebody, oh, how do we learn with two senses? Or what happens when you combine smells and colors? And these are things that are not necessarily very dense with jargon, with technical jargon. So conceptually, it's not very difficult to explain. And I think that helps make it more sort of palatable and relatable for a broader audience.
Yeah, I agree. And it's something we always kind of think about and commonly say, commonly it's been said that it is that way, but the actual real hard data, was kind of not there. So this is really wonderful. And I think something almost everyone is interested in, because it's, yeah, something, when we make memories and they're maybe many, many years old, and all of a sudden we recall them and people think about that, I think, a lot. So
I
yeah,
was even thinking,
oh, go ahead.
this is almost everybody has had, where let's say they smell a cleaning product and then kind of they immediately see in their mind, they're transported back into time where they were visiting their grandmother's home, or I don't know, they were in school. So it's really interesting how that seems to be a very universal and relatable phenomenon.
Yeah, exactly. That's yeah, I think it what for me personally as like an immigrant kid since always, like since I'm little, was that the minute you're, let's say, in your home neighborhood, like you arrive, you feel like you never left. No? I don't know if anyone experiences that.
Yeah, definitely. We are both
Like
immigrants.
all the
So
time in between is gone. Like it's so
yeah,
interesting.
exactly. Yeah.
Okay, I think we can start. Thank you for this really put it into context pre-discussion we had. I think that was actually really helpful. And so, yeah, I want to welcome everyone to Science Society. We'll start with introductions. So welcome really to the both of you, as we said, that are willing to, use this different platform, make an account and everything. We all really appreciate it. So welcome, Dr. Okray and Pedro Jacob. And yeah, let me give the audience a short introduction first, and then we'll get into interviewing the two of you a little bit. So Dr. Pedro Jacob joined the University of Oxford in 2017 as a postdoc in Scott Waddell's laboratory and in the Center for Neural Circuits and Behavior. And he did his undergrad degree from the University of Lisbon and his doctorate from the University of Cambridge, where he worked in the lab of Bertold Hedwig, studying the neural circuitry evolution of the song system in crickets, which is by itself really interesting. Maybe one day we invite you back to talk about crickets. We had a really interesting couple of spider rooms, so I think crickets would be wonderful too.
Yeah.
And your main research interest now is in behavioral flexibility, how it arises and evolves, and how dynamically changing environments demand that animals constantly adapt their behavior and update memories and incorporate new information. And Dr. Okray, you did your education at the University of Chicago in biological sciences, and then you did your master's degree at the Catholic University in Leuven Belgium, and also your PhD. And then you worked there until 2016, and from 2017 till now, you're at the University of Oxford. So, welcome to both of you. And yeah, the first question that I would like to ask you is, how did you discover this passion for science, or that you wanted to, have a career in science? Was it a childhood dream, or, something that came later on with a class or teacher, museum trip, whatever it is we think it's really interesting? And would ever like to start, please go ahead. Thank you.
Thank you so much for having us. And yeah, again, it's a pleasure to speak to you all. It's difficult to answer this question about, how how did you decide to be a scientist? Because it felt sort of an organic path for me. I think one thing that I do remember as possibly a big incentive was that I used to be crazy about museums. So I didn't have a biology museum close to where I grew up. I grew up in Turkey, actually, close to Ankara, the capital. And however, there was a geology museum. And I just loved visiting this museum, and basically kind of made friends with the curator there. And they got help. They sort of let me handle some of the specimens. So I got into sort of understanding what science is a bit like from the physical sciences side. And then in school, I realized that I was very interested in sort of living systems, and how, especially how evolution works, was very interesting to me. And when I started my university degree, I developed a passion for immunology of all things, which then changed into neuroscience. But overall, I think I've always been super fascinated about sciences.
Thank you for inviting me. Me, I think was I guess how every little kid gets into science by watching nature documentaries, and I've always told my parents that I wanted to go and be a kind of a David Attenborough type thing, look at animal behavior. I've always been fascinated by animal behavior, and during my degree, I was fortunate enough to be serendipitously put in a class of animal behavior, and then that refueled my passion for neuroscience and like science in general. So it was basically I was always fascinated by animal behavior and how does it evolve, and yeah, that's it.
Yeah, that's really interesting. That you both said that it was not necessarily in school itself, but outside of school. That
Yeah, definitely. I think this is a testament to having exposure via media of documentaries, and now I think the quality of those have improved enormously as well, which is really nice. And for me, museums were a whole thing. I remember the first time, I was much older then, but the first time that I saw the Natural History Museum in London, I just was under its spell for days. Like I completely lost my mind over it. So for me, museums were a big thing.
yeah, that's really wonderful to hear, especially for people that are giving money to museums and so on. Um, and also for this documentaries that people are making them. I think it's very encouraging to keep financing them and have them available for everyone, ideally for free. And I agree, it's such a wonderful, different way of learning that probably evokes some sort of, I don't know, emotional experience combined with, gaining knowledge that is unbeatable, probably.
Definitely.
Yeah, it's a
Definitely.
wonderful story that you both shared. And I think it's really interesting because a lot of, speakers answer these type of questions, sometimes similar, sometimes not. But a lot of people say that, I don't know, a trip to nature or living close to nature or museums and so on were kind of important. So yeah, it seems to be very, very, very important for children's lives. And then coming to the research we will talk about today, how did you start working on it? How did it come together? Was it very hard and difficult, or is there any kind of story around it that you would like to share with us? Thank you.
Yeah, so we would like to, by the curriculum, the CV that Katarina presented you, you see that you started in the same year, but what's not there is that we start 8 days apart.
6 days apart.
6 days apart, because you appeared on the first day of the year, the 1st
Yeah,
of January.
I traveled on the 1st of January because it was cheaper. So I I just showed up at work on the 1st of January, and that's what he's making fun of.
Yeah. So we initially didn't work together, but we had common interests. Zainab was always interested in like how, because we're going to talk about this later in the talk, is that we use a very simple from that was developed in the 1970s by actually Ship Quinn, that is the postdoc supervisor of Scott. It's a very simple old paradigm of this T-maze paradigm, but Zainab, she can correct me if I'm wrong, but she was always interested in understanding, like, this is a very simple setup, and animals don't experience this simplicity in the environment. And I was fascinated by how animals adapt to different contingencies, so how they adapt to changing environments as well. So this was kind of the start that Zainab can tell a bit more.
The size of. So actually, yes, we didn't start working together, but we've been good friends for a long time. However, so when I first started in Scott's lab, I didn't have experience doing behavior, and I started. I arrived and I wrote my a project to get funded, which got funded, but then the project didn't turn out to be viable. So I was basically just trying different things, asking different questions. For about 2 years or so, I kind of floated. And then while I was doing experiments, one of the things that I realized using this T-maze setup was that the flies that we were trying to teach associations to were learning more cues than what we were explicitly teaching them. What I mean by this is that in this T-maze paradigm, we train the flies to associate smells with a particular reward or punishment. And I also, what I did realize is that they were also learning something about kind of the chamber they were in, the texture of the chamber, perhaps the color of the chamber. And that if I kind of replicated this chamber between the training and the testing phases, that this actually improved memory. So this is something that's well known in the learning and memory field, that if you match the encoding and the retrieval phases of which is like the learning and the memory retrieval, then you get better performance. But nobody really understood why this happened in the brain. And when I actually observed this, another thing I realized is that we could call it context, right? So the chamber that the animals were in, that they were learning in, But this context is a very ambiguous word. So, I had to really delve into literature starting from like the 50s, 60s, 70s to try to understand, what is known about the so-called contextual memory and what is context anyway. And context is essentially sort of this a complete collection of both internal and external cues that the animal uses to understand its environment. And what I realized is that, for us to start probing the neural basis of contextual memory, we need to reduce it to in its dimensions and choose, let's say, one very explicit additional cue and see what happens, what kind of interactions happen between two different sensory cues while the animals are learning to associate these cues to a punishment or reward. So I picked basically odors. So I picked smells, which is what we've been using, and I added colors as a specific contextual cue. Does that make sense?
Yeah, it's really interesting that you chose smell and color. I think I find it a really interesting combination. And this is a great segue to your actual talk. So I think we can dive into that now. So everyone, feel free to access the slides that are pinned on top of the room. And yeah, the stage is yours. Thank you so much.
I can also add, like, basically, it was an interesting story of how me and Pedro started working together as well. Because once I decided that okay, I'm going to try to teach the flies with two cues, both of different senses, both smells and colors. I designed some experiments and was optimizing these protocols. So here I am, and one day it worked out actually quite well. I got excited, trying to replicate the results. At some point, the phenotypes that I was observing just disappeared. So the setup stopped working. And I was just going crazy. I couldn't figure out what was going on. And I'm like, I started almost not believing it myself. And because I was trying, couldn't recover the phenotype. And one day we were in the fly room, which is like where we keep and work with our flies. And Pedro was, sort of going along with his own work. And I was on the verge of tears, almost being like, I can't take it anymore. I can't take it. I need to I either figure it out or I quit now, right? So that I go and do something else. And, Pedro being the gracious, wonderful person that he is, I said, you know what, just let me give it a try. Just another pair of hands. You never know. So I explained to him how to do the experiment, and he actually did the experiment, funny enough, in another room, which turned out to be the problem. Apparently our humidity sensor was off, right? So something so small can have such a huge effect. But Pedro did this experiment in another room and recovered the phenotype, the behavior that I had initially observed. So I was extremely excited, and we very quickly discovered that we work very well together. And this whole story that we'll be telling you about today is essentially us working super closely together to the point where almost all of the experiments we split half an hour. So, I would start the experiment, and a few hours later, I would just call Pedro, and he would just take over, like almost like a relay race. So, in this sense, this is truly the sort of product of a very collaborative effort.
That's wonderful to hear. And it's really interesting because it's the second like room and where this was pointed out by by the researcher, because we had here last week Dr. Hertz, who discovered, together in a collaboration, uh, the full structure of the nuclear pore of the cell. And this was like over 20 years effort. And the main thing he kept pointing out was how important it was that all the groups, because it's impossible to do by itself, how all the groups kind of work together over time and were like family, and they could all trust each other and work well together, and without that would never happen. So thank you for pointing that out again, how
Definitely. I mean, I have to say, I think this is something to me that is just as important as the discovery itself, which is to really, it's not necessarily always a very easy thing. We always say, oh, we should collaborate, as a team, but there are elements of the system that sometimes make this difficult, right? And, we're essentially in a meritocracy. There are sometimes resources that we all compete for. But what does collaboration within this boundary look like? Because, I've got to experience, I used to always be collaborative, but I've got to experience collaboration. I've never experienced collaboration at this level before. And now I realize the true power of it. It's more than just an additive thing. It's very synergistic, because we're both humans, right? And we were able to sort of carry each other through times that were difficult, that were frustrating. So we just moved with enormous speed and momentum. So I sort of urge everyone to kind of try to discover what that would look like in their own life and in their own work, right? It does take work, I have to say. It's not always, you have to communicate very well, you have to be very open-minded, you have to understand each other and empathize with each other, but it's definitely something that I think is very, very worth it.
Yeah, thank you for pointing that out. I agree. And then the other type of rooms we had sometimes lately was where the level, like where the additional technology came into play to, use human collaboration with AI to. It was about figuring new steps to develop certain materials and semiconductors, new type of semiconductors, and so on, which in the future will add another interesting layer to it. But I think, yeah, human collaboration is really it's wonderful when it happens. So yeah, so well, so thank you for sharing that.
Great. So we can go ahead and talk to you about our work then.
Yep, please go ahead. Thank you.
Sure. If you open the deck, that the PowerPoint slides, this is, we didn't really know how this was going to be. So it's a bit of a mixed up deck of slides. But if you go to, say, slide 4, or even slide 3, where this is basically just some results from a human study that they did, where basically also, while we all know that learning using multiple senses improves both the acquisition and the recall of memory, there's actually, people have done quantitative studies and shown this at the data level. So here you can see that if you learn using both audio and visual cues, you first of all learn quicker, and then you remember more correctly over time, compared to, say, learning with visual alone. So we wanted, while this is well appreciated and also appreciated by educators, for example, who come up with teaching strategies at school, we don't really understand the neurobiological basis of how the brain does this. So this was essentially what motivated us to really understand the circuits and the neurological activity that could explain this phenomenon. So we work with the fruit fly, Drosophila, and this is for various reasons, but the main reason is that, this is, by the way, in slide 4, which is just the title slide. But this is because these animals, they're essentially like, they have brains that are numerically simple relative to higher animals, but they're able to generate a surprisingly complex behavior repertoire, including some quite rich learning behaviors where they associate cues with meaningful events like a sugar reward or a punishment.
So if you see in slide 5, this is basically what I mentioned in the beginning, that this protocol, this paradigm was developed by Shipquin in MIT, in MIT, where he teach flies how to associate either a sugar or a shock to the flies, and this is what is called conditional stimulus plus. And then we have here we show it as odor 2, and then we have another odor, odor 1, that is the unpaired odor that we call conditional stimulus 1, conditional stimulus minus, sorry. And then after some amount of time, we can test the memory for the preference between odor 1 and odor 2. Here you can see it's a very simple paradigm with only odors. And then if you move to slide 6, we can see that if we pair this protocol with sugar, we can see what we call a positive performance index. So basically, we calculate the number of flies in the conditional stimulus plus, the CS plus, arm minus the conditional stimulus minus, the CS minus arm, divided by the total number of flies, and we have a performance index that in this case will be like 0.2 or something. So positive would mean approaching the CS plus, and negative would mean avoiding the CS plus. And one thing that is good to point out is that this is a population analysis. So each dot that we are going to present in the behavior data set is always comprised by about 200 flies. So it's a very robust phenotype, very robust phenotype. And so the story is about multisensory. So we saw that.
If you also turn to slide 8.
Yes. The story is about multisensory. So what we did is that we adapt the same paradigm, but now we added a color. And what we did is that, like you see, other 1 associated with color 1 and other 2 associated with color 2. And then again, after some time, we test for the preference of the 2 different combinations. And in this case, what we're going to focus is mainly on, you could see in the paper, we have both punishment and reward, but here, just for simplicity, we just going to focus on reward or sugar memory, what we call sugar memory. So if you move to slide 9.
Yeah, so here what we did was to essentially design 4 initial protocols to test different aspects of multisensory learning, and also just to see, okay, can the flies actually learn both the visual and the odor cues in this particular setup, because people hadn't combined these 2 before. So the first one that we tried is the visual only learning. This is where we only presented colors during the training, and hope that the flies could associate, say, the sugar with a particular color. And during testing, we would only use colors again. Then we have the olfactory only condition, and here we train the flies to associate sugar with an odor, and then we test the preference to this odor, and during testing, we only present the odors. And then we have the congruent condition. This is where we paired colors and odors, during training and during testing, and we kept these particular odor color pairings consistent between the training and the testing. And finally, we added another condition, kind of initially as a control, but we called it the incongruent condition. This is where we would pair odors and colors again, but we actually inverted the odor color pairs between training and testing. So this is in slide 9. Again, sorry, apologies that we keep on forgetting to mention the slide numbers, but it's essentially going in sequence. So we went ahead and if you actually remove can you actually, Katerina, can you see the full graph in the deck that you have?
Yeah. The visual, the first three are kind of a little bit less like opaque, but we can see the full all three.
Okay. Okay. Yeah, I think this was basically since we didn't know that we wouldn't share our screen, we kind of left it as a PowerPoint presentation. But if you can see the data, then that's all you need anyway.
Yeah, but if you go to slide 10, maybe you if you move to slide 10, what you can see is that a visual memory, right? Like the V, a graph with a V, right?
Yep.
Exactly.
Yep.
Yeah. But nothing else.
Yeah, exactly.
Is that correct? Okay. So, is there any way to put it like in slideshow mode in this deck?
Yeah, so if everyone wants to use it as a slideshow, please open it if you have in Google Slides and then press play. Basically, then you can have it as a slideshow if people have.
Because that will because we have the kind of the flow of presentation. So sorry about this. So basically, but I will, for the people that can see, great. But basically, what we saw is that if we present the visual condition only, we see that we couldn't see really a memory, and so basically they wouldn't have a preference between CS plus, CS minus. So and next we try to if you see in the slide mode and if you press forward, we try to present the odors only condition, and we see a performance index that is normal to what we see before in our normal unisensory experiments. But if we combine the odors and the colors in the next, if you move the slide forward, uh, the next, the congruent or the C condition, you can see a massive increase in the memory performance, and that is was what is really exciting, that we could see that even flies could associate to cues, and this would get a performance much better than just the unisensory cues. And if you could see from the data, is that is not additive, because we couldn't see any unisensory visual information, so it's not additive. There is something else going on the system that shows this enhancement in memory performance. And interestingly is that we found, if you move the slide forward again, if you move the slide forward again, we could see that in congruent condition, we found that if we switch the pairs of colors and the odors between training and testing, we see that actually the memory performance in incongruent or I is much lower than the unisensory olfactory or DO condition. And this is kind of interesting because it would show that the flies are now exposed to a conflict between two sensory cues, and they actually perform worse than before. So now if you move to slide 11, I'm sorry about the hiding in between slides. So now if you move to slide 11, we kind of try to test now two different paradigms. Is that what happens if we want to know is the memory improvement would be apparent if we retrieve the multisensory memory using only one modality. So if we can retrieve the original memory just by presenting either odors or colors to the flies, and see if this could recapitulate the enhancement that we saw before. Is this clear so far? You can ask questions along the way.
Yeah, and please ask us anything that's not clear, so we can just make sure that we explain it better.
Yeah. So in slide 12,
Yeah, so in slide 12, you can see the results of these experiments where we train the flies using both visual and olfactory cues, but then we retrieve the memory with only part of this multisensory stimuli. So we would retrieve the memory only using odors or only using colors. When we when we train the flies multisensorily and retrieve the memory with odors only, we saw that the memory was still much better than unisensory, so olfactory only learning. We thought this was absolutely fascinating, and we realized that it works the other way around. So, for example, when we trained flies with both colors and odors, but then retrieve the memory using colors only, the memory performance was still better than if the flies only used visual cues, which basically in this setup didn't really produce a strong memory at all. So we were very encouraged by this and started to wonder, okay, can we understand the neural basis of how this enhancement works? So we took a look. If you open slide 13, we decided to focus on the mushroom body, which is this structure, L-shaped structure in the fly's central brain, which we consider to be the center for associative learning and memory. And we know actually quite a bit about this structure based on decades of research by both our group and many, many others. And if you go to slide 7, go back to slide 7, you'll see a simplified schematic of the mushroom body. So here, what you can see is that the mushroom body, this L-shaped structure, is formed by the so-called Kenyon cells. These Kenyon cells are essentially cells that carry sensory information. They encode for the identity of the sensory stimulus, and they extend their processes, in this case axons, to form this L-shaped bundle. And these axons of Kenyon cells are innervated by dopaminergic neurons, which relay value, right, valence. And for example, in the case of sugar, they're activated by sugar reward. And then in these dopaminergic neurons, when they're innervating the axons of Kenyon cells, they form very distinct compartments. In this particular slide, the gamma-5 compartments we highlighted because this compartment is relevant in sugar learning. And then finally, there are the mushroom body output neurons. These are neurons that ultimately direct behavior of, let's say, avoidance or approach. And the whole learning system at this level works such that the Kenyon cells the particular Kenyon cells that are active during stimulus exposure. If this activity coincides with the activity of certain dopaminergic neurons, this changes the plasticity, the weight of the synapses and the plasticity of the connections between Kenyon cells and mushroom body output neurons, which then later on skews the behavior towards approach or avoidance. Does that make sense?
Yeah, it's really interesting to see. Thank you.
So this is, you know what? I mean, of course, this is a much more simple. We've simplified it quite a bit because there's, this is a network that's highly interconnected. There is some interneurons which we won't get into today, which we worked with as well for our paper. But it's a really neat system of a relatively manageable number of neurons that are able to function in sort of different permutations to create all sorts of learning phenomena. And it's really interesting to study these to get an idea of the logic of how neural circuits work to create memories. And of course, people ask, is this the same in humans? And what we do know is that a lot of the principles that we've discovered in flies, especially in like associative learning, these principles are conserved in the mammalian brain. And yes, of course, the mammalian brain has many layers of added complexity, but at least the way that the rules and the principles of how it works seem to be conserved. So as I said, we focused on the mushroom body, and in the mushroom body, we focused on these Kenyon cells that, if you turn to slide 14, we focused on these Kenyon cells that receive visual inputs. So these are the gamma D in yellow and the alpha beta posterior Kenyon cells in pink. And we mostly focused actually on the gamma D, and that's what we can talk about.
So this we take advantage. We take advantage of this gamma D and alpha beta posterior because also what we have in the slides is that we have a full reconstruction of the central brain and now also the ventral nerve cord, so like the motor output region. So we know basically how each neuron is connected, and what you can see in slide 14 is a reconstruction of these Kenyon cells done by this. It was done by Google machine learning that reconstructed all these neurons. So now we have access to where these neurons are connected and which are the input and output of these neurons. This will be important later on in the talk. So if you move to slide 15, we also take advantage of the years and years and years of tools that were developed in the Drosophila field, and we can basically silence or activate neurons that we are of interest. And then in this case, what we did is to use a temperature control block of the activity of these neurons. So it's a mutant for the dynamine. So it doesn't allow the exocytosis of a neurotransmitter. And basically, what we can do is that we can block these neurons at any time point that we want, and then see their effect in memory. So this is what is exemplified in this.
Katerina, one question: what kind of a time frame are we talking about? Because we're trying to explain the details, but we also don't want to run too much over time.
You're fine as long as you have time. We have time, so please
Okay.
feel free to. Yeah.
All right. So we'll try to. Yeah. Well, hopefully we won't keep you here for too long. But basically, indeed, as Pedro said, we made use of these genetic tools that we have that allow us to very tightly control neuronal function. So what we did was that, okay, we first wanted to see, here we have the visual Kenyon cells. Let's try to block these visual Kenyon cells and see, like, okay, are they involved in at least the visual components of our multisensory learning paradigm? You would expect that they would be. So we block them, as you can see on slide 15 again, we block them only during the testing to see if the memory performance in all these conditions were affected. Again, this is the congruent condition, the visual retrieval, the olfactory condition, and the olfactory retrieval. From here, basically, if you go to slide 16, you see that the yellow and the pink are when we blocked either set of the visual Kenyon cells, the yellow being the gamma D Kenyon cells, and the pink is the alpha beta posterior Kenyon cells. And when we blocked these during the testing phase of the congruent condition, we found that the enhancement of memory performance was gone. So this kind of made sense because we thought, okay, we're blocking the visual input. So, the animals can't rely on this to improve their memory. So that made sense. And then we also found the same thing for the visual retrieval. So this is where we would train the flies with odors and colors and retrieve the memory with colors only. So if we blocked visual Kenyon cells during the retrieval, we completely abolished the memory, which made sense because, the retrieval only involved colors. But then what we found very interesting is on slide 17. This is where we blocked the visual Kenyon cells in the olfactory and the olfactory retrieval condition. It's important to note that the testing phase of these conditions only have olfactory stimuli, so they don't have any visual input. So we reasoned there should be no reason for the visual cases to be involved, at least in the olfactory, right? And indeed, in the olfactory only learning, you can block these visual cases and it doesn't seem to affect the performance at all. However, what we got very excited about was that when we blocked these visual Kenyon cells, in this case, specifically the gamma D Kenyon cells in yellow. So when we block these cells during retrieval, where there's only odors, this actually really significantly impaired memory performance. So somehow the visual Kenyon cells became required doing the testing to retrieve the full multisensory memory, even though they were retrieving it using odors only.
So in slide 18, so we wonder why is this visual Kenyon cells like the gamma D are required for olfactory retrieval of multisensory memory. So what's happening at the level of these cells to allow this requirement? So what we did on so you can see it on slide 19. In slide 19 is that what we did is that try to look at their activity pattern after we train them, either with olfactory only, like the gray squares there, or the congruent, where we show both olfactory and colors. And then after some time, like 6 hours, we test them under the microscope using a voltage reporter. And this is important because normally what we do in the lab is use GCamp or these calcium reporters. But the thing that we'll see on slide 20 is that flies, if you look at the first set, the top set of the slide, the olfactory condition, is that these flies actually respond to odors by being inhibited. So we record in this specific region that Zeynep mentioned, the gamma 5, and we see that in purple, in purple dark, in both purple and dark and light purple, it's the response of a fly to the average response of the flies to both the CS plus and CS minus. And what you can see is that in gamma 5 region, we see an inhibition to the odors. And this has already been observed before, that actually these visual Kenyon cells respond by being inhibited to odors. But we tried initially to use these calcium reporters like GCAMP, but the problem is that it's very difficult to observe this inhibition. So to observe the like a change, you can see a change in inhibition, but we never sure if this is a disinhibition. So if the response is less inhibited or actually being excited. And so what we change, like I mentioned, what we changed to ASAP is that we are actually seeing an excitation. So if you see the bottom part of the panel in gamma 5 region, we see that the CS plus, the dark purple curve, responds with an increase in activity in comparison to the CS minus that is still inhibited on the olfactory retrieval.
So just to add one thing to that, is that remember that CS plus is the rewarded odor and CS minus is the non-rewarded odor. And the gamma 5 region is the region where this region is relevant for sugar learning. This is where the dopaminergic input that relays sugar information goes. And one of the things that one of the nerdy jokes that we like to make is that, you know, now we're seeing if we actually train these flies with both colors and odors, then these previously visual only Kenyon cells now become excited by odors. And we always say that this is equally exciting to us as well as these neurons. That's why I just wanted to plug in this this very nerdy joke that I'm very proud of at the moment.
Yes.
That was an important interruption to the program.
So, and then if you can see on the right hand side of the slide, you see that we also image a completely different region on the polar opposite of the mushroom body, what we call the gamma 1. And in this case, we didn't see any effects in the activity patterns of these neurons. So we are recording the same neurons because they bundle from the region where they have the cell bodies, and there is basically a track that runs from gamma 1 to gamma 5. So they have a track, and so we record the same bundle of neurons both in gamma 1 and gamma 5. But why don't we see an effect in gamma 1 is because this is the known place where the punishment dopaminergic neurons innervate. So basically, because we are using sugar, there is no reason why these neurons should change their activity pattern, because there was no dopamine being released in this area. So that's why we don't see an effect in this area. And this was some sort of control, an important control to have, that it's not like a generalized sensory increase, but it's very specific to the compartment where dopamine is released.
So one addition to that is that indeed, as Pedro said, we weren't expecting this excitation in the gamma 1 because already from our results from gamma 5, we had the intuition that this excitation was only happening because there was dopamine in there. Why did we know this? We knew it because the CS plus, it was only the CS plus odor that was actually eliciting an excitation. As you can see, the light purple data does not show this excitation. So there's something about having both colors and odors as well as dopamine that was making this sort of change, drastic change in responsiveness.
So this is in slide 21. this is basically a very simplified schematic of what we're thinking that was happening. So basically, like we said in the gamma 5, and also we know gamma 4 is also, we were just simplified, but we know that gamma 4 is also received reward dopaminergic neurons, reward dance. And what we think is happening is that when the odors and colors come during learning, or in the left-hand side column, what we call multisensory repetitive training, what we think is that there is a change in the synaptic connection between the Kenyon cells and the output neurons, the Kenyon cells and these dopaminergic neurons, and they change their activity. Then, when we present the CS plus odor, like the conditional stimulus plus the reward odor after training, there is some sort of activity that is also only change on that side of on the gamma 4 and gamma 5 part, and not on the other region of the gamma D canyon cells. So this is what we mean by these little arrows going from the odor to the from the gray arrows going from the CS plus odor to the yellow arrow.
And at that time, we didn't really, we didn't know anything about what could be bridging this activity. So, at that time, we were just thinking, oh, the activity is literally looks like it's jumping from one stream to the other, only in this compartment. So there's some invisible bridge that forms between these two streams when dopamine is present. So we then set out to identify what that bridge could be. But before that, actually, what we didn't mention is that on slide 22, we also checked whether the directionality of that cross-modal activity was the same from Kenyon cells that responded to visual stimuli to odor cells. So in this case, visual retrieval. If you remember, we also had this experiment where we trained flies with odors and colors, and we retrieved the memory with colors alone, right? And we saw that this also elicited an improvement in memory performance. So we thought, okay, is it going to work the same way? Can we train again, like multisensory, train these flies and then give them color and see whether the response, we could also observe a response in their olfactory cells, which would normally not respond to color. And we won't show you the data for that, but indeed this is the case. And if you do want to see that data, that's on slides, the setup is on slide 24. And slide 25 and 26 is where that data is. So we had to change the setup a little bit under the microscope setup, because we had to figure out a way to deliver color and as well at the same time record neuronal activity, which kind of the channels that we would record that overlapped in wavelength with the colors we were giving. So we had to kind of come up with a nifty way to separate that. But if you want details on this, we can explain later on, or if you ask about it, or you can take a look at the paper. So as I said, we were interested in this bridge, which again, you can see in slide 27. So this bridge that would connect the two streams, the visual Kenyon cells and the odor Kenyon cell streams, especially where dopamine was. So what we found is, you can continue.
So what we know before is that there is these two massive interneurons in the fly brain called DPM, the dorsal paired. Oh, sorry, in slide 28, there is these two massive interneurons in the fly brain, in the fly mushroom body. The DPM, the dorsal paired medial neuron, that here colored in blue, that is serotonergic, and there is this anterior paired lateral neuron in purple that is GABAergic. And we know that these neurons are crucial for olfactory learning. And what we could see in slide 29, is that the we first focus on finding the anatomical evidence to support the idea that that these neurons could be involved in this change of activity in the Kenyon cells. So APL, we have some data on the APL, the anterior paralateral neuron, but APL being GABAergic, meaning that it's inhibitory, it couldn't be the candidate to be the reason why.
The source of my excitement. It couldn't be inhibitory, definitely.
It could, because we are seeing an excitation. So we focus on the DPM, the serotonergic neuron, and try to find this if there is any anatomical evidence that DPM bridges the excitatory activity between the different Kenyon cells. And like I mentioned, now we have these connectomics, like these inputs and outputs of these neurons. So we could just see if there are basically a match between where these neurons input and output from. So what you can see in this figure, in figure 29, is what we call a dendrogram. So it's basically the neuron kind of flatten out, and you could see the different regions that we see here, marked gamma 1, gamma 2, gamma 3, gamma 4, gamma 5. Both the ventral and the dorsal stream of this neuron. And what we show is that there are DPM synapses in close vicinity of visual, the gamma D Kenyon cells, that mark in yellow, the yellow dots, and the olfactory Kenyon cells, the gamma M Kenyon cells that are marked with gray dots. And what we argue is that these are in close proximity enough to allow this kind of microcircuits between the two components to exist in the learning relevant compartments.
So one thing that I want to add here is that our study is a good example of how useful connectomics data can be. So what I mean by this is, again, connectomics data is basically the data that we've gotten from electron microscopy reconstructions of the brain, which have been traced to reveal the individual synapses of each of these neurons in the mushroom body. So what we did was that here we have the problem of seeing, okay, we're looking for a bridge, something to connect the activity between the two Kenyon cell streams, right? There could be different ways that this happens. For example, one hypothesis that we considered was that these two Kenyon cell streams could be directly connected, right? Why not? They're essentially in a bundle, so maybe they have synapses that just relay information from one stream to the other. But when we looked at we were able to eliminate this hypothesis just by looking at the connectomics data, because we saw that there was literally not enough synapses that connect gamma M, so the olfactory Kenyon cells and the visual Kenyon cells, to account for this activity. But what we could see, as Pedro just explained, is that, okay, if we add this DPM into the mix, then we see that the DPM is anatomically suitable to connect these two streams in a robust way. So, I mean, connectomics is awesome. And this is Dr. Niels Otto who actually led this analysis. So if you look at slide number 30, and we're coming to the end here, DPM was becoming sort of our prime candidate for accounting for the excitatory connection between these two streams. So we wanted to see whether at the behavioral level we could prove its involvement. So we blocked DPM function in the same way that we blocked these like visual neurons in the beginning, and we blocked it during testing again. And then what we saw is that, first of all, if you look at slide number 31, you can see here that if you look at the right-hand side, you'll see the olfactory only learning, and blocking DPM during testing does not significantly impair or affect the performance, the memory performance in olfactory learning. However, when we looked at multisensory learning, and especially the olfactory retrieval and visual retrieval conditions, where we train multisensorily and retrieve with one modality, when we block DPM, we actually abolish the multisensory enhancement. So this kind of solidified the idea that DPM, if you look at slide 32, that DPM is actually the connection, provides the connection between the odor coding Kenyon cells and the visual Kenyon cells.
So this is what we mean by this kind of this pathway between the CS plus odor in gray, and then we have this pathway in blue, that is the DPM neuron, leading to an increased activity in that particular yellow branch of the gamma D Kenyon cells. So when when this odor and these colors were associated during training, the presentation of either one of the modalities, because we have the same data for the inverse, the activation of either one of the modalities activate the other modality via this route. Yeah.
Yeah. So I guess like the final piece of data that we can talk to you about is the bonus appearance of the 5-HT2A receptor. So this is slide 33. This is a serotonergic receptor that's conserved in flies as well, so it's in mammals. And it's a particularly interesting receptor because it's known to be the target of hallucinogens. So things like, for example, psilocybin. And what we did was to hypothesize that, in this model where DPM connects the two streams, and the end of that connection, so when DPM is carrying over the activity, for example, the gamma D cell, the Kenyon cell, should receive that input, which is serotonergic from DPM, via a serotonergic receptor. So we screened basically all the serotonergic receptors that we know are expressed in these cells. And we know this also because we have access to a single cell transcriptomic data. What we found is that if we knock down the expression of the 5-HT2A receptor in these visual Kenyon cells, we actually can interrupt the multisensory memory enhancement. And we also saw that our imaging data recapitulated or strengthened this result. So this is slide 34. And what you can see here is that we compared basically the olfactory responses after multisensory training in gamma D, in the gamma 5 compartment, in animals that had 5-HT2A expression in the gamma D cells, and on the right side, flies that did not or had this receptor expression knocked down by using RNAi. And what you see is that when we knock down the expression of 5-HT2A, we actually abolished this cross-modal activation. So we prevented the ability of visual Kenyon cells to get excited by odors.
And one interesting thing as well is that we also, besides 5-HT2A being the receptor for hallucinogens, we also know that neuropsychiatric disease like autism and schizophrenia have been shown to have deficits in multisensory learning, and this has been associated to the serotonergic dysfunction as well. So this was a nice
parallel.
A nice parallel,
yeah.
exactly. That in flies we also see an important role for the serotonergic system to be involved. And then if you move to slide 35, it's just basically the summary, and basically what we show is that multisensory learning in Drosophila improves memory even when single modalities are tested, and this is what we kind of associated also to what happens in the hippocampus with this pattern completion. So where one stimulus is enough to recall the whole memory. Also, we show that multisensory training binds modality-specific neurons and expands the memory engram. So the engram is like the cells required for memory. So here, our hypothesis is that the memory enhancement that we see after multisensory is just basically because we have more cells coding for this memory, and via this bridge between cells by DPM, and the binding is dependent on serotonin and dopamine signaling as well. And finally, on slide 36 is basically the people that contributed to the work.
Yeah, this is a team effort, like more than the two of us, of course, as well.
Yes, and then two incredibly gifted undergrads, Chiara and Kieran, a PhD student, Paula, Cliff, that is our engineer in the department that help us with the imaging setups, and Niels was the person responsible for the connectomics.
He's our connectomics wizard.
Yes.
And on the right is basically kind of all our
except
me. Yeah, it's the rest of the lab, which of course, we often have discussions with, and their input was invaluable. And that's it. Thank you so much for listening to us. If you have any questions about any part of the work or any part of, our day-to-day life working on this, so anything, we'd be happy to explain.
Yeah, thank you so much for this wonderful presentation and for guiding us through your data. It was really wonderful to understand and to follow this basically.
Yeah, we hope it was clear.
Yeah, and while I was following along, it was it was really interesting to hear you about what you predicted, and especially the to make then the connections what you predicted, what the data was, and how you followed along. It was really interesting. And I'll ask a few questions, and then I'll give Dr. Shah and Denise and anyone that shares in the chat maybe a question, the opportunity to also ask a question. So please, everyone, if you have questions, either share them in the chat or raise your hand to come here to ask them in person. And this is as I said, this is such an interesting story, and how this different visual versus olfactory system kind of get involved and are cross-modal. Basically, I thought this was a really interesting result, because I kind of always thought before, like back in time, that the cortex of modern mammal cortex, there it's very easy to think how they can be cross-modal. I don't think back in time we suspected that from different systems in the Drosophila. So I thought that was really interesting. And from an evolutionary perspective, since I think Pedro is also interested in that, do you think that the more complicated, basically, the environment and like also social environment became, this was kind of the first step of going into having a modern cortex? The modern cortex is kind of an algorithm to be, it looks very similar no matter if you look into sensory motor versus, prefrontal cortex, anatomy as you couldn't, if you would cut it out, put it somewhere else, it would be really hard to distinguish. So, do you think that that came next out of necessity, and this was kind of the first step towards that?
Yes. Thank you for the question. Yeah. It's definitely like, of course, flies are much simpler than mammals, and they don't have the cortex. But what we found is that indeed these animals like flies can do these associations and can bind this information and basically use the same molecules that we see that are important in mammals. So we suspect that based on our previous work that in the lab, that we show that similar parallels between vertebrates and flies, the principles are basically the same. They're just like expanded in the cerebral cortex of mammals and primates and humans, but basically the rules are basically the same, and that's the beauty of using the fly, that we can actually probe the rules that are happening, and where it's much more difficult in vertebrates.
Yeah, I would bet, for example, don't hold me up on this, but it's very likely that serotonin and even 5-HT2A would be responsible for sort of connecting the different streams of information in the mammalian brain, even connecting the different cortices.
And yeah, that's really interesting. Would you, since the GABAergic system kind of had not just an expansive role, but still a role in there, would you kind of categorize that the GABAergic system is more kind of to separate things and serotonin to connect and collaborate in the brain, or is that way too, for the broader audience?
No, I mean, to be honest, I think this is a pretty, in my opinion, it's a good sort of general categorization, right? So I think at least in the fly, what's happening is that indeed DPM is sort of providing this excitatory glue or excitatory bridge, but then the fine tuning, the restriction of this excitation specifically to the relevant compartments by responding to dopamine is probably happening via the GABAergic inhibition. So that GABAergic inhibition is fine-tuning the excitatory input to restrict it to the relevant locations.
Yes, and we also in the paper we also have some data that supports this by blocking a specific dopaminergic neuron in this GABAergic dopaminergic receptor in this GABAergic neuron. We see an effect in the memory. So
Where is it?
uh,
It's somewhere in our data. Oh, slide 45, I think that's APL. I see it.
yeah. But it doesn't matter so much if you go back to the slides or not. But what we saw is that if we block this dopaminergic receptor in the GABAergic neuron, and we do olfactory or olfactory retrieval, we abolish the memory. So they need this APL is basically the source of localizing this increase in memory enhancement to the specific region where dopamine happens, where dopamine is released.
And I don't know if in Drosophila is there one system a little bit faster than the other? Like, is the visual system a little bit faster than the olfactory one, or is it the opposite way? I was thinking if that may play a role, or to which one becomes then more crucial for retrieval, or adapts faster, that it becomes crucial for the system kind of point of failure for the system if there's a multisensory memory going on.
So, I mean, I think if I understand correctly, first of all, what do you mean by faster? But or are you asking in general, for example, the bidirectionality? You know, is there one sense that's dominating over another in this setup? Is that the question?
Yeah, dominating and has kind of a faster information processing, like, you know, in mammals. For example, the auditory system is the fastest one to arrive. Like if there's a threat or so, they're the least connections that need to be bridged. So if you want to learn the fastest, basically, you just have a few neurons to kind of startle like some animal. So and then the visual system is also really fast compared to like other sensory systems. You know, that's why people theorize you have kind of this weird fears that you don't know where they came from. It's because you didn't even consciously had time to process them in type of a threat situation, and then you're scared of red and you don't know anymore that you saw maybe something red there because it was too fast. Is there something similar like that in the Drosophila, and is that why you know, there's kind of a difference between the olfactory and the yeah.
Yeah, thank you. Yeah, so one thing that maybe wasn't clear when we talked, that we are actually, so we are of course visual visual information comes from the eyes, right? But here we are looking at the mushroom body, where basically we kind of know that they have basically both the olfactory streams and the visual streams project to these Kenyon cells, and basically they have the same information time. So they are not, basically they're not faster because they are exactly in the same
location.
They are exactly in the same location, and this is not the primary, like the visual Kenyon cells are not the primary visual receptor. So these are just cells that exist in the mushroom body that also receive input from the visual system. And like the olfactory Kenyon cells are not the primary cells, so they are just cells that receive the sensory information. So basically, we don't assume that they have a different one being faster than the other, but we have some data that suggested that there are some sort of combination of this, and some of the senses, especially because if you see the T-maze schematic, it's basically a tube where the flies are kind of bound together. So it's difficult to assume that it will be different if a fly is flying and they have they are much more visual information coming through. So basically, we are bound to the paradigm that we're using, and it's not like necessarily the most ethological relevant one, like flies walking around.
Yeah, I mean, one of the things that's important to note is that we're doing a very unnatural activity for the flies, right? So this is not in any way recapitulating something they would experience in nature. And also, we have this word called salience, right? We don't know what how these cues are received by the fly. So we don't know, whether being in a wind tunnel, having these colors would be like sort of less distracting to them, or would they pay more attention to odors? We can't really understand that. But as Pedro said, by looking at the mushroom body, we're kind of assuming that the encoding of visual and olfactory stimuli are happening at the same time. There might be very slight differences in, the temporal resolution of it, and we don't know about that. But what we do know is that we do have an experiment where we did not, I don't think we included it in the paper actually, But if we present, for example, the colors and the lights in a sequential way with sugar, so not together at the same time, but sequentially, this doesn't seem to improve the memory as it does when we present both the colors and the odors at the same time.
And like if you see the data that we present, we don't see really a visual memory alone, right? So we assume that this wind tunnel or this image is more the olfactory information will be predominant. But would assume if we put them in a fly mode, like I mean the flight mode, so where the flies have to learn how to associate cues and turn around while flying, maybe the visual system there takes more predominance because the visual system is much more important to orient it themselves than the olfactory system. So I guess it's also the paradigm
that you use. Yeah.
Exactly. Yeah.
It's really interesting. There was a paper years ago, which kind of reminds me. So I don't know if you know the paper where there was shown that visual rhythm perception learning in humans was better if
Yeah,
there
I
was
know the
auditory.
paper.
Yeah.
Yeah, I know the paper you're talking about. Yes.
Yeah.
Mhm.
Do you think there's a correlation there? Like this is kind of the underlying mechanism. Like you already kind of alluded towards that, but I thought it was interesting to make that connection there.
Yeah, I mean, there's actually what we did not mention is that in the mushroom body, at least for flies, right? There's no reason for us to think that this is a special, this is a mechanism that's special only for the connection between olfactory and visual streams. Because in the mushroom body, there are also, for example, cells that encode for gustatory or tactile cues. And we would assume that the mushroom body would use similar mechanisms to sort of parse together this information as well. So I don't see, we can't really say what happens in humans, but we would imagine that the brain likes to sort of utilize mechanisms that work, right? And it doesn't reinvent everything from scratch, but sort of does variations on similar mechanisms. So I imagine that there's something perhaps similar going on.
Yeah, thank you so much for that. And I wanted to hand over the microphone to Dr. Shah. Hi, Dr. Shah. Nice seeing you.
Hi, Katerina. Hi, everyone. And thank you so much for sharing your wonderful research with us. So my question is about, because you mentioned about the dopamine receptors, and specifically when we are talking about the midbrain, and we are aware of the Drosophila and the cellular diversity that we have there, and specifically when we are talking about 5-HTP, serotonin, or dopamine. I was just wondering to ask you, uh, is there any kind of information that you could gather about the histamine? Because when we are talking about monoamines, this one can as a role, and I was wondering to ask you about it.
With that, we didn't specifically look at any other monoamine, and but we know that is a neurotransmitter in the photoreceptors, but we didn't look at this activity as well. We didn't, yeah, because we know that.
I don't think there's people haven't described histamine in the mushroom body, so I'm not sure. It's not one of those. Um, it's not one of those neurotransmitters that have been in the sphere of learning and memory circuits. So we didn't really look into that because we were focusing on where this particular interaction could happen.
And the duration of the learning was 6 days, right? Based upon your experiment.
No,
How
it's
much was the duration?
6 hours, actually. 6 hours. And we
Okay.
did test different time points. So what we did show is that all this sort of multisensory interaction and the memory improvements we could observe at immediate memory. So this is right after learning, at 6 hours, which is kind of like a midterm memory, and at 24 hours. And we also saw that the same mechanisms were at play in aversive learning. So this is like, for example, punishment learning, and again at different time points. So it seems to be a relatively general mechanism for associative learning with multisensory input.
However, when it's optimized on 6 hours, it seems, and we are dealing with the postsynaptic, correct? Mostly because we are talking about dopamine, and I'm assuming that more
Can you
than.
speak up a little bit? We are having difficulty hearing.
Can you hear me now? Better?
A little bit louder if possible, sorry.
Okay, so when we
Yeah,
are talking about dopaminergic, we are focusing mostly on postsynaptic, right? And I'm assuming when we are talking about Drosophila and midbrain, more than 70% supposed to be postsynaptic, correct?
Yes, yes.
Yes. And when we are just want to compare that to the learning center, so I'm assuming it's supposed to be post to the learning center, correct?
So what is supposed to be?
Location, learning
so,
center in
so
comparison.
the dopamine essentially is being released on in the compartments where axons of Kenyon cells, right, are synapsing with mushroom body output neurons, and the presence of dopamine changes the weight of these synaptic connections between the Kenyon cells and the mushroom body output neurons. So in that sense, it is postsynaptic.
Yes.
Correct. Okay, so thank you so much.
No, thank you for asking.
Yeah, thank you. And Denise, did you have a question that you wanted to ask or comment?
Thank you Katarina. Selam Zeynep Hocam. Ben Deniz, memnun oldum.
Selamlar. Ben de.
Hi Jose. Thanks for both of you for this really interesting presentation. I didn't really understand some of the finer points of how different neurotransmitters will be implicated in these sorts of learning or memory. And it was just when you were the whole premise of this reminds me of proprioception in orbit of Earth, and it's not really a memory recall, but it has to do about the integration of sensory inputs, multimodal integration. So, for example, if you're in orbit, if you are inverted in 0G, you'll become disoriented, and one of the reasons for that is you're not getting any sensory input from your lower extremities, and the full integration of those sensory inputs will give you the ability to, for example, not fall down a flight of stairs because you haven't miscalculated the distance and the range and your rate of speed, that you'd have to do that safely. But it was very interesting about the color and odor. I didn't necessarily think about how these two things interact. When we're talking about recall, I feel like smell is one of the easiest ones for people to understand. You think about what's cooking in your grandmother's kitchen, and sometime you may walk past that smell and it brings you right back to that point
Exactly.
in time. It was really interesting about the sugar water reward. I was curious about, in a world with no budget, what would be the next reward circuit that you might want to test outside of sugar water?
So using flies.
Using flies, or any other organism you can think of.
So I think we've been looking at different reward pathways in our group, right? This is something, for example, we know a little bit more about how water reward works. We know a little bit more about how, for example, sexual reward works. We have some other things in the pipeline as well looking into this. But Pedro, for example, is getting interested in how addictive or compulsive circuits work. So in this case, I don't know if you would call it reward, but would you call it reward?
It's at some point it's also reward, because in a basically OCD is basically obsessive compulsive disorder. You get a reward or a relief from executing a compulsive behavior. So in this sense, yeah, there will be a reward component, and we know that dopamine is also involved in this. So yeah, I guess that's
Yeah.
yeah.
And Dennis, thank you for letting us know about that. I didn't know about that mismatch in, with the gravitational force. If you couldn't have sensory input from your lower extremities, that kind of messes with your perception. That's very cool. And what we do know is that, brains have evolved to process information by integrating information from different senses, right? Like the representation of events and objects in our mind are inherently multisensory. And insects, for example, bees use this to, when they're flying around, they use both, for example, the color and the smell of flowers to guide their behavior, and they learn much, much quicker. And for us, it's basically the examples are completely, they're infinite. Essentially, I don't think there is a situation where we rely on a single sense.
And that's before we even get into the social learning aspect
Yeah,
of bees
especially
and
emotional
conditioning.
learning aspect as well, because these are also internal cues. We're only talking about sensory cues here in our example. It's external sensory cues, right? So you have to realize that this also, there are numerous internal cues that the brain needs to integrate, essentially. Sorry.
Yeah, fascinating. And it's would you would you say that that is a lot of the unlocking of certain functions or restriction of other functions by just hormones and the chemistry, or are there other things that I'm missing?
Yeah, I think that's exactly it. We are like unmask basically what we. Of course, it's a bit of a far stretch, but we are basically giving flies synesthesia. So they're like by modulating this dopaminergic and serotonergic system, we are basically the flies are basically experiencing colors or the color memory by using olfactory neurons. So we are basically unmasking this connection by modulating these neurotransmitters. Yeah.
And it's interesting to consider, this thing about hormones or the state of the animal also plays a role. For example, the very basic one is that sugar memory in flies, the expression of sugar memory and the retrieval of sugar memory is dependent on the fly being hungry. So the fly actually will not express a preference for a previously sugar paired odor if it's not hungry. So there's something interesting
Fascinating.
with respect to. Sorry?
Fascinating. That's I don't know. Like
Exactly. And
dealing with the flies in Turkey, it just it seems like they're hungry all the time.
and I mean, I think what I'm also now interested in the future is to sort of delve deeper into how the same stimulus, essentially, the same sensory stimulus can elicit completely opposite behaviors. So this can happen, right? So if you
Aversion, like in
anthropomorphize it,
humans with alcohol aversion.
yeah, or for example, if you see, let's say, a tiger in the wild versus in the zoo, you might have sort of very different
Completely,
reactions to it,
yeah,
right?
completely different
That
reaction.
exactly. So there's something interesting about, this is obviously a very complex behavior, but the good thing about working with flies is that we can sort of be super reductionist and try to bring it down, strip it down to its basic level, where we say like, okay, how do you make the simplest experiment for this? And what does that tell us? Which is how we went about doing this project that we just presented to you.
I was that was so interesting. I was curious about if there was anything that was out of scope of this paper, but you still wanted to talk about it maybe, like there wasn't any room for it, but you still feel it deserves to be talked about. We did a bunch of that right now, so I'm very happy about that already.
I'm thinking, for example, with the 1 second, we're trying to. How did you fix this last time? Okay. We're still trying to navigate Clubhouse, but now I think we got the hang of it. I'm trying to think if we did anything that. What we were kind of interested in knowing is that there are some counterintuitive findings in our work, which is Pedro alluded to this a little bit, where he said, we still haven't fully demonstrated why the memory is improved. So we know, it's implied from our work that indeed you expand this engram, so you're basically storing this memory in a greater number of cells, which then should drive a stronger response. But we don't really know what's happening downstream of these Kenyon cells and how these so-called mushroom body output neurons are interacting perhaps to improve behavior.
And also because we didn't mention this because it was too complicated to go in fine detail, but we know that at least in olfactory memory, the rule of learning is a depression between dopamine and the output neurons and the Kenyon cells. So we have an inhibitory effect from dopamine. So, but here what we see is an excitation. So the learning rules that happen after multisensory are different from the learning rules that are happening during unisensory memory retrieval. So that, I guess, that's the big question marks now, is like, what are the exact learning rule that's happening in this
And what he means by learning rule is basically at the microscopic, well, everything's microscopic level here, but at the microscopic level of the synapses, right? The specific, like biophysics of the different molecule changes and the sort of activity signs, right? They at times don't match up in the way that we expected. So there needs to be, like, we would want to, in an ideal world, if when we had more time and didn't have to move on, that we would look into this.
Wow. Yeah. Okay. This was a really great conversation.
But what was interesting, Denise, about this, that when you said that, this is the only time where we thought there was a counterintuitive thing. And it's the first time, I've been in the lab for 20 years almost now, and it's the first time that I've been working on a project where literally every single experiment that we did was in line with what we thought was happening, like in the way that or what things started fitting together one after the other, that we didn't have one piece of information that was really out of place until the very last bit, which the project gifted us with like one open door where things didn't really fit. But until then, it was really fascinating because I've never had this where things just work out one after the other. So it was very exciting to work on it.
Wow. You think that's just a function of you having looked at this information for 20 years, or is there something else at play?
No, I mean, I haven't worked on this for 20 years. I'm actually, I've been working on behavior only for 6-7 years now. But I don't know why it worked out like this.
I think what our lab, since Scott's run this lab, it's basically we are very focused on this area, so on this mushroom body. So we really know how learning and what are the learning rules and what are we can almost kind of predict what happens in different paradigms that we didn't test, because we actually know what are the connections, especially now with all the connectomics and transcriptomics that we have, like these new tools. We can actually go at the single genes and single neuron interactions, and this makes it much easier to, I guess, to predict what is going to happen. Mm-hmm. Yeah.
The moving parts, the number of moving parts are much, much, much less, right? I mean, I sometimes, it's not the best comparison, but it's a little bit like having four box Rubik's Cube versus a giant, giant one. So we have something a lot more manageable. So we're just playing with the fundamentals and are able to ask big questions and do manageable experiments that give us quantitative answers. And this is the beauty of the fly. You know, people don't like working on the fly. They kind of think it's, well, some people don't, but the first reaction is almost like, oh, do they even have a brain? But in fact, there's a lot that these animals can tell us, because it's almost like a little playground in which we can run simulations almost.
Do you get that attitude more from lay people or other scientists, that the fly is such a basic organism that it has nothing to add to knowledge about humans?
I think lay people are more surprised that the flies have brain, but I think the aspect of the flies that don't have anything to offer to humans is, I think it's more an attitude from scientists, I guess.
Yeah, it's getting better, to be honest, but unfortunately, we are still a little bit, researchers that work on animals that are so-called less than mice, right, or rodents, invertebrates especially, like we're a little bit the underdogs. We have to always still justify why the work we do is relevant for science in general. And now, however, if you think about it, we were looking it up the other day that I think there's like 10 Nobel Prizes in total, or was it 6? Did I just?
9.
Oh, 9. Sorry, 9 Drosophilists have won Nobel Prizes for their work in Drosophila. So this already tells you that these are extremely powerful research models.
And
Yeah, I
they're easy
wanted
to
to say
execute.
like how memory also works was not done in mammals, and a lot of things we know how neurons work, how information processing works, like the synapse works was done in squid, like giant axons, and
Yeah.
Candel, that was all, not mammals. A lot of things we know about how cells work are from yeast, and
Yeah.
most of it's pretty much the same, even DNA wise. Like there's such a huge overlap. So yeah.
Yeah.
Definitely. I would have loved to study microbiology, but then I fell in love with flies, so that it's fine. But I would never leave this model organism actually, because for me, I think Eve Marder, I don't know if you know her, but she's like she's a superb neuroscientist in Brandeis. And basically I once heard her on a podcast say something like, as scientists, we need to choose where in the spectrum of ambiguity we want to be. For example, if you study human brain, you have to tolerate a huge amount of uncertainty and ambiguity and complexity, because that's just the system that you're working with, right? You can ask very interesting questions. You can ask questions that pertain to language, for example, which are specific literally to the human race. However, for more general or more universal cognition questions, you can also use systems like the fly. And in the fly, I like sort of it looks almost childlike, but I like the simplicity of it. And within that simplicity, there's still a huge amount of complexity. So we're talking about like 200,000 neurons in the brain. That's in the little worm, the C. elegans, there's what, 300, I think? Yeah. So Yeah, 390. So this is, that we all, if we as scientists kind of like understand our model well and define what level we want to be at, and then actually communicate across these dimensions, then that's how we get the most progress.
Yeah, I couldn't agree more. And I think it's really interesting. You went in, you went in the discussion even how the serotonin receptors that mediate this multisensory binding, and how these are targets of hallucinogenic drugs that are really studied a lot right now in addressing mental health illnesses. So, this is just a very detailed but an example how the overlap, um, you know, is it just an example how these systems overlap, and even things like very specific experiments can make this connection to things we thought that were just humans that would experience, like having this hallucinations and these connections that usually the brain doesn't do, and then have this visual perceptions and auditory perceptions, which I think is really interesting to make that connection, because it's really a lot of visual and sensory inputs that make this weird connections in the brain, and drive this hallucinations. And how do you think that this research, like in the future, your research model could make us understand better these systems, and also then disorders that are related to it, such as schizophrenia and autism on both ends, basically, of the spectrum? How do you think that research could be really lead to understanding these disorders?
So I actually, by the way, it's funny because I was just Googling whether anybody gave psilocybin to fruit flies. It
What's
turns
the
out that
answer? I'm so curious.
so, Scientific American, Katrina, I'm going to get back to your question, but Scientific American just has an article from 2018 saying that magic mushroom drug evolved to mess with insect brains. And then turns out that a single dose of psilocybin has long-lasting antidepressant-like effects in fruit flies. So I haven't read the paper, but it seems like people have been trying to give psilocybin to fruit flies. It would be interesting. We'll check it out later on.
I was
I
wondering if
think
that would pass an internal review board, and apparently it has. Amazing.
it is. So actually, I did my PhD in a department that focuses on neurodevelopmental and neurodegenerative diseases, and I used to work in flies then as well. And I even wrote a review based on, how can the fruit fly, what can the fruit fly tell us about, human disease and neurological disease, essentially. And again, it goes back to what we've been rabbiting on about, just going on and on about how these fundamentals and the rules and the principles are conserved, right? So I think in this sense, there's a little bit of disservice done by fly researchers ourselves by, trying to claim relevance constantly by saying like, oh, okay, the depression model of fruit flies, or the, I don't know, just pick your model, like the Parkinson's model of fruit flies. Of course, there has been extremely valuable research in this, right? But we need to be very careful exactly what we're describing. What does a model for a human disease look like in the fly? Well, it literally means, we just want to understand the biology of what's going on more. And we can sometimes screen drugs very effectively because these animals are very easy to rear. So if we have a general idea of the biological mechanism, we can also sort of use it as a platform to screen for drugs that modify it. And people do this even in the yeast. So we don't have to go at the level of flies. But I guess what I'm trying to say about this is that we do have quite a lot we can learn about disease using flies, but the real, let's say, the real money is our discoveries based on the basic science of like what is the basic mechanism underlying all these disorders.
Yeah, and basically of course we can't, like, it's difficult to have a schizophrenic fly or autistic fly because these are very human phenomena that have like social interaction, and it's a and basically we can't ask a fly that if we heard smell colors, or like we have to rely on the behavior. But what we have in our work is that we have a mechanism that we think it's how multisensory learning happens, and we've been seeing that humans also rely on serotonin and possibly on this 5-HT2A receptor. And so that's the contribution maybe to is that is this serotonin doing this bridge to allow multisensory learning in humans possible, and this is this bridge or whatever kind of um, parallel to this bridge that we have in the flies in these disorders as well.
So it brings ideas to the table. I think that's where the value of it is.
Yeah, thank you for for sharing that. And yeah, I agree. It won't get us, to the behavior and so on, but basic cell mechanisms maybe, and a connectome insight would give us a lot of information that we couldn't as easily or at all gather in other systems. So
And
I
in
think it's,
genetics as well. Like genetics wise, there's I think there was something like 79% of disease causing genes are conserved in the fly.
yeah, I agree. Yeah, I think, and also gene expression modifications due to the drugs and what they do then to the cells and the gene expression would be really interesting to study in this model, and in this model of having this multisensory memory formation and recall, and to see how these drugs kind of change that would be really interesting.
Definitely. I think both
So
me and Pedro, not to make this longer, but both me and Pedro are. I'm speaking on your behalf, Pedro. Please correct me on this. But one of the reasons why I want to stick to Drosophila is that now, for example, I want to jump to other big questions, right? So of course, there's still a lot more to figure out with this particular paradigm and this multisensory learning. But I've already moved on in my mind to all sorts of other questions. Like I'm curious about the immune system. I'm curious about basically how, let's say, yeah, again, going back to contextual memory, but how the immune system might play a role in shaping our perception and cognition. So that's the cool thing about working with flies. You kind of can move quite drastically from one question to the other.
yeah, I agree. It's really, and having you have also way more animal available and way cheaper
Yeah,
than
and
save.
you don't need to get the ethical consent that much, although we try not to sacrifice our animals for nothing, but it's definitely not at the level of other animals.
Yeah, I mean, your research opens up so many new avenues of what people can do next. So this is another really beautiful thing about your research, and I'm really curious to kind of learn what comes next also out of your collaboration that you explained was so crucial, and you work so well together. So we will be curious to follow, your collaboration along, and yeah, maybe you'll come back and share some more about it. And thank you so much for, staying here for these 2 hours and, making the count and everything. It was really a wonderful and really interesting discussion. So we are really thankful, and I hope you enjoyed it. Because
Yeah, no, it's been amazing. Thank you so much for showing interest.
yeah, thank you. And I hope, the nervousness about using this platform now turned into it was okay.
No, no, we are fine.
Yeah.
And
Now
it's
I'm
good to not
basically,
have a video, right? For 2 hours, if you have to sit there
oh,
like
yes, it's
looking
crazy.
good.
Yeah.
You're more relaxed, I think. Yeah.
Yeah.
Exactly. So yeah, thank you for sharing your work, and I good luck for everything. We hope you'll get a lot of more funding in the future,
Oh, thank you so
because
much.
yeah,
Yeah,
it's
we
really
just hope
interesting.
that we're both going to be on the job market. Well, we are on the job market, just applying for group leader positions. But I just hope for, at least on my behalf, it was just so much fun. It was a lot of hard work, so don't get me wrong, and a lot of drudgery, and it was exhausting. But at the same time, my God, it was so much fun. So this is all I dreamed about as a kid, to be able to be in this position of making discoveries. And I really hope that I understand that the system, our funding system and our scientific discovery system puts us sometimes in very difficult positions. But I hope that for all of us that we can continue holding on to that feeling, and not have very, very long deserts of sort of being stuck, let's say, because it's a really fun feeling to be able to discover things.
Yeah, I agree. It's fun and really hard, and I hope in the future, hopefully one day it will get less hard and more fun, the whole system
So
for us.
I
Because
don't know
I
how.
kind of feel once you get the group leader, the most work is bureaucracy and more bureaucracy. So I hope that won't be the case for you.
I was
And
just
so
The admin, you mean, right? Like there seems to be a big admin. So I'm usually very interested in the sociological sides of this as well. So indeed, being a group leader, it's very different depending also on the position you have and also on the country that you have as well, I think.
yeah, and the institute you're in, the department, how heavy it puts burden on people to have all this administration
Yeah.
stuff. Like it yeah, check for that. I can only give you advice.
Of course.
The
And as
more
women, we tend to, like, they call it the unpromotable work, right? So as women, I think we're particularly vulnerable to get sucked into these sort of committee work. And, well, they can be quite rewarding. Like, I completely understand what you mean.
yeah, it depends where you are. I don't want to get too much into it, but yeah, because I don't know everyone's system, of course, and just yeah, and then if you have in the department more people that are kind of in your area of research or not, I think also makes a big difference. If most people work on humans and you're kind of almost the only one not, there you have to go through all this administration loops that is more designed for humans, which are way more complicated, and you have to go through them anyways. Also, like it depends really on the school and how the
Yeah,
department
exactly. Yeah,
works and so on. So
yeah, yeah, exactly. Yeah.
Yeah, but it's also exciting. So I think that's the cool thing about this type of work is that life always changes,
Yeah.
and the things we do, we have to learn always change. I think that's exciting. So yeah, we are really looking forward to follow you along and your next
Thank
steps,
you.
and maybe you'll come back and
Thank
share
you.
with.
And there's always something. So I do have a theory that at least maybe it's a way that I console myself, but I do have a theory that there's interesting questions in almost every sort of corner you look, right? This can be in this capacity of being a scientist, or even if you work in other jobs. I think there are ways to kind of make it more interesting in general. So I'm hoping that, if academia works out, fantastic. But if not, I'm sure eventually I'll end up doing something cool. We'll see.
Oh yeah, for sure. You will, and yeah, whatever will work out. Will be. We can turn, I think, everything into something good and interesting. So, well, first of all, congratulations for what you have achieved
Thank
together,
you.
Thank
and
you.
Yeah,
yeah,
it's been team.
yeah, it's happy. We kind of get the privilege to get to speak with people when they achieved, the sort of, when they achieve this and they're happy about their work. So we are very
Yeah.
privileged in that way. Maybe we should move on to like middle stage to get a more to get a more appropriate reflection how life is in science. But
Yeah. I see what you mean. Yeah.
yeah.
I mean, to be honest, there have been days where, like, I think I sort of was so frustrated that I just stopped everything and went home crying, basically.
Yeah, that's what I mean. That's part of it, and
And there's no guarantee that it ever works out. So that's the catch. Like, I completely understand that, this paper, while we're so happy it worked out, at all levels, it could have just not been possible. And just because
yeah,
of some sort of luck factor.
yeah. There's always the luck factor that people under appreciate, and yeah, people shouldn't be discouraged if something they're working on doesn't work out. It happens,
Yeah. Yeah.
and
Well,
also to
it's
colleagues
still
of
difficult,
mine.
but yeah.
Some things just don't work, just don't get funded, and then
Yeah.
you have something else. The best advice I think somebody gave me when I started my PhD is have a main project, but also have always one or two side projects that you can switch to if necessary,
Yeah,
if
Yeah.
things don't work
definitely.
out.
You can procrastinate on one using the other one.
Yeah, this is something that Pedro does well. It's true.
Exactly.
So you can break the monotony by just working on multiple problems at the same time without diluting yourself so much. And one thing that I do is actually I keep a little notebook as well, where I just write down random questions and things that interest me. And sometimes I go back to it because, there comes a time where you have to sort of generate, as you guys know, you have to generate these questions completely by yourself. So, they don't just descend on you on the spot. Therefore, keeping kind of like a non-judgmental catalog of all these questions is really helpful as well.
Yeah, and I think what helps in kind of an environment if there's kind of a judgment-free lab meetings between labs sometimes, and then people
Yeah,
come
yeah.
up with questions that would help you with kind of judgmental free. I can only encourage, PIs and so to keep it that way. Because if you have an PI that I had for a while that says, I don't want here people to come with data and they don't know at all how to interpret it, just don't
Uh-huh.
give that burden to the audience. I don't think that was a good idea. It made a lot of pressure than people not sharing things with fear of, I think it's a good thing. Like the group can together think about, answers questions. I think that can only be a good thing. I don't know how you think about it, but I would encourage judgmental free meetings in that department.
And also, Katrina, it's wonderful that you started the Science Society. I mean, from what we understand, it's a really fantastic way to talk about science. I really enjoyed it.
Oh yeah, thank you. Yeah, that's most of the time the feedback. Like in the beginning, people are very nervous because it's different and so on. But then people say, "Oh, it's really interesting," and we really cover a lot of different fields of research. From archaeology, imaging techniques, nanotechnology, I don't know, space, black matter, like dark matter, and to biology and environment and neuroscience, like everything. And it's really interesting after a while, since we've been doing this, how people from different backgrounds come up with really interesting questions that make these discussions kind of different from like a regular conference meeting. I think that kind of what got me hooked when this app kind of started. And it was during COVID times, so everyone was at home anyway, so people had kind of time to do this. But that's kind of what I think was interesting to keep going on and investing time in.
Yeah, it's really interesting.
Well, thank you for being part of it. And yeah, maybe we'll hear you again and see you again. And feel free, everyone in the future that is listening, reach out. If you still have some questions, reach out to me. I'll try to answer them. And yeah, thank you so much. And I hope everyone has a great rest of the day, morning, evening, wherever you are. So thank you.
Thank you very much.
Thank you very much. And
Thank you.
very nice to meet you all.
Yeah, nice
Bye.
to meet you. I'll close the room in 3, 2, 1. Bye, everyone. Thank you.